Great. And all right. So thanks everybody for joining in. I'm sure there will be more popping on as we go. So welcome to this Pediatric Tech Talk. For those who are unfamiliar with the series, it's organized by the Children's Healthcare of Atlanta Pediatric Technology Center and the Georgia Tech Pediatric Innovation Network. And we are here to bring conversations with key investigators at Georgia Tech about pediatrics and healthcare research and initiatives that join the pediatric healthcare space. And today we have a real treat. So I'm here in place of Leanne West, Chief Engineer of the Pediatric Innovation Network and the Pediatric Technology Center. Leanne is on a plane somewhere, but I'm delighted to step in. I'm MG Finn, Chief Scientific Officer of this organization, and the real star of today is King Jordan, who you see. So King is a professor at Georgia Tech in the School of Biological Sciences and director of the Bioinformatics Graduate Program. He has a PhD in genetics from the University of Georgia, which we won't hold against him, in 1998. Was a staff scientist at the NIH for several years, and then started his academic career at Georgia Tech about 17 years ago, and rose rapidly through the ranks and has established really a nation and world-leading program in bioinformatics. He is also directs and is the inspiring leader of an effort to build biotechnology capacity in Latin America, which is a wonderful initiative that I doubt we're going to have a chance to talk about. And instead, King is going to give us, I think, a bit of an introduction and some issues that he sees as important in the area of genomic medicine and health disparities. So King, welcome, and why don't you kind of introduce yourself and what you do?
Sure. Thanks a lot, MG, for that introduction. So I won't cover that ground again, but I'll just jump in and tell you right about my research. So I have a computational biology laboratory lab in kind of air quotes, because a computational biology lab is not a wet lab that many of you may be used to, but rather just a room full of computers. So we use computers, but I'm really a biologist and a geneticist by training, so I use computers to ask biological questions. And the questions that I am particularly interested in and motivated by now, or at this time rather, have to do with human health. And I got my training in genetics, but everything about human health and really everything about our phenotypes, what makes us who we are, is a combination of genetics and the environment and the interactions between genetics and the environment. So we use bioinformatics, algorithms, tools, software, data, databases, and recently population biobanks, which are these massive collections of data from participants who donate biological samples, which are typically used to characterize DNA and other biological molecules, fill out lots of surveys and questionnaires and give demographic data, social data, data about diet and lifestyle, and very importantly, grant access to their electronic health records. So this is an amazing combination of genetic, biological, demographic, social, environmental, and health outcome data. So we analyze those data computationally in order to understand why people get sick. What are the risk factors that increase individuals' likelihood of getting sick? And in particular, we're focused on health disparities. Health disparities are differences in health outcomes between population groups. Population groups can be defined in lots of different ways, but we work mainly in that capacity with NIMHD. NIMHD is the National Institute on Minority Health and Health Disparities. They are particularly interested in health disparities that impact minority, racial, and ethnic groups. So we're looking at differences in health outcomes between racial and ethnic groups in the United States and other countries in the global north, trying to understand how genetics and how the environment contribute to those disparities and hopefully tease them apart. And the last thing I'll say is that we look at all of that along the axis of genetic ancestry. So race and ethnicity are socially defined, they're social concepts. They are related to genetic ancestry. Genetic ancestry, on the other hand, is a characteristic of the genome. The story of our ancestors is written in our DNA. So we hope that we can use genetic ancestry as an analytical tool, a means by which to disentangle or decompose genetic and environmental contributions to health disparities.
Amazingly interesting. So do you focus on any particular types of disease or looking at health kind of writ large? And if it's the latter, like how do you, what's the metric? What do you look at? Lifespan or other things? Yeah.
So it's more of the latter in the sense that we are data scientists, right? So we go big. We're data scientists. We like to go big. So we start with a population biobank, and we do what's called a phenome-wide analysis. So a lot of people have heard of genome-wide analyses, but what a phenome-wide analysis is where we look at the whole phenotype, the whole phenome, which is a collection of all phenotypes. We're interested in health-related phenotypes. So we use electronic health records to characterize health outcomes for literally thousands of diseases. Since we are interested in disparities, we analyze all those thousands of diseases, but we're looking for diseases that satisfy some criteria, that check some boxes that we're interested in. So first and foremost, we're interested in common diseases. These are diseases that have relatively high prevalence within and between population groups, so they have a big impact on public health. And we're looking for disparity diseases, diseases that show differences in prevalence among groups. Now, having said that, MG, one disease that we have converged on with our collaborators at NIMHD, which checks those boxes, is type 2 diabetes. So the prevalence of type 2 diabetes is really increasing. It imposes a very heavy burden in terms of health, but also on the economy, morbidity, mortality. But it also has very different, big differences in prevalence between groups. So in the US in particular, African Americans and Hispanics have very high prevalence of type 2 diabetes compared to the majority white group. So type 2 diabetes is not only a common disease, but it's a complex disease. And that means that it has a multifactorial causality. So there are lots of things that go into why, again, that central question of someone's risk of type 2 diabetes will be higher than someone else's. Genetics is important. It has high heritability, but quite obviously the environment is important, diet, lifestyle, socioeconomic factors. So type 2 diabetes is a really interesting kind of model system for us in the sense that it has a very high overall prevalence. It has big prevalence differences between groups, and it is multifactorial with contributions to risk of disease from genetics and the environment and their interactions.
And how are these kinds of insights, for example, in type 2 diabetes, how do you see them being used? Are you trying to create uses? Like, what do we do with this information, or what do health professionals or organizations, what would you like to see happen, or what is being done with these insights that you're developing?
Yeah, that's a great question, right? So, and this is one of the fun things about being a scientist at Georgia Tech. You know, I just as a quick aside for the group here, I started really as a basic scientist. So I was not a health scientist. I was a basic scientist doing basic genetics research. And just by accident, my work kind of migrated over to the health field. And so that means that I am very much a junior epidemiologist, right? So, or a neophyte, right? So I'm learning epidemiology all the time. And one of the things that I've learned is that for epidemiologists, the gold standard in terms of risk factors are modifiable risk factors, right? So you said, what can you do with this information? And what we want to do is identify those risk factors that we can modify such that we lower individuals' risk of disease. Now, that's kind of really far from my research. I'm still doing basic health research, not really clinical research, but we still want to keep that key paradigm in mind. Now, that raises a kind of interesting and tricky issue, which is that genetics, now this is leaving aside CRISPR-Cas9 and the revolution in genetic editing that is kind of on the horizon. If we leave that aside for the moment, we all have the genetic variants that we're born with. Of course, we accumulate some new variants over our lifetime via somatic mutation, but those variants are written in the DNA of our 37 trillion cells. So they are largely not modifiable. So then the question from an epidemiological perspective becomes, well, why would you be interested in genetic risk factors given that they're not modifiable? But what the hope is that by looking at how the genetics and the environment interact, particularly as it relates to differences in disease risk across groups, we can then use genetics to lead us to modifiable risk factors, and in particular, modifiable risk factors that may be more relevant to one group or the other, to help us really hone in on where it is that we need to look at for specific groups in order to lower the risk of disease for those groups.
Not to ask you to put something out there that you were uncomfortable, but do you see any particular factors for type 2 diabetes that you would advocate as to change for those who are at risk? Or like, what pops out at you if you're at that stage yet to say, hey, let's pay attention to this factor?
So there are, of course, the standard things. So type 2 diabetes is essentially a disease of excess blood sugar, right? So you get insulin regulates the amount of sugar in the blood. So we eat food, we convert that into sugar. We use that sugar to, for energy, to power our cellular metabolism. But the modern diet is just absolutely loaded with sugar, as we all know, right? So we're kind of almost assaulting our bodies and our cellular systems with sugar all the time. Now, insulin is responsible for converting that sugar into a form we can use, but as we're assaulting our bodies all the time, we get insulin resistance. So that's where the insulin is no longer able to convert that sugar, and it accumulates in the bloodstream. Now, I say all that to say that there's very obvious things that we don't need our research for, like diet and exercise, right? So we know what those things are, but having said that, it can be difficult in this modern environment to control those. Now, as it relates to disparities, one of the things that we've become very interested in, and this is a little more subtle and complicated, at least for us, is the relationship between socioeconomic deprivation and those diet and lifestyle factors, particularly as it relates to disparities. So different groups in the US have different levels of socioeconomic deprivation. And what's really interesting, in socioeconomic deprivation is a risk factor for type 2 diabetes, and it's a risk factor for those diet and lifestyle modifiable factors that, again, increase your risk of disease. Now, you can measure socioeconomic deprivation at the individual level, but you can also measure it at the neighborhood level. So there's this interesting concept called ZNA, right? So we've all heard of DNA, and the Z in ZNA is your zip code, right? So your DNA is important for your health, but your ZNA, where you were born, where you live, your local environment, especially as it relates, again, to socioeconomic deprivation, is very, very important for health outcomes, including but not limited to type 2 diabetes. The complexity is that when it comes to minority, racial, and ethnic groups in the US who suffer a disproportionate burden of type 2 diabetes, individuals who have the same level of socioeconomic deprivation when we measure it at the individual level in terms of their income, their education, their access to healthcare, and so on, tend to live in neighborhoods where the place-based metric of socioeconomic deprivation, the ZNA, indicates more socioeconomic deprivation. So that was a quite striking and unexpected finding that we made. And so now, how does this relate to, you said, well, how can you use this information? Now we know that for individuals from different groups, their relative levels of socioeconomic deprivation at the individual level may tell them something different about how that relates to risk of disease, right? And that can relate to surveillance and early intervention, and just keeping a close eye on individuals who, if they were members of one group, we might think, well, they really have a low risk of disease. But given the fact they're members of a minority group who, again, at the same levels of individual deprivation, tend to confront a neighborhood environment with higher levels of place-based deprivation and more local risk factors, might have a higher risk of disease. So that's an example of how we can kind of use that information to tailor interventions at the population level. Having said that, I should mention this interesting concept called precision public health. So I think most people are probably familiar with precision medicine or genomic medicine, where we use information about individuals. I mean, precision medicine writ large is just using data to tailor health interventions at the individual level. And genomic medicine is the genomic version of that, whereby we're using information about individuals' genomes to tailor their healthcare. Precision public health has a different level of intervention. It's intervention at the population level instead of the individual level. So the idea there is that you're looking at characteristics of populations in order to intervene at the population level, at the level of public health, which is more screening, early intervention, and hopefully prevention, again, based on characteristics that are distinct to different populations. And that's where the health disparities piece comes in.
Yeah, I hadn't heard that before. That's cool. So connected with this kind of issue, you mentioned to me before, an issue facing health disparity research is a genomics research gap. That was a phrase you used, and I wasn't familiar with that. What is a genomics research gap, and what are its implications with respect to health equity?
Yeah, this is a really, I'm really glad you asked that, MG, because this is a fundamental challenge to genomic medicine and to health disparities. But at the same time that it's a challenge, it's also an opportunity. So I mentioned before, and I'll just repeat, that genomic medicine is using individuals' genetic information to help make better informed decisions about their healthcare in terms of disease diagnosis, prognosis, but even therapeutics. All of genomic medicine is built on this big foundation of basic genetic research, right? Where we look at genetics and we try to associate the genetics of individuals and populations with their risk of disease. The genomics research gap refers to the fact that the vast majority of individuals who participate in those studies, it's something like 80 to 90%, have European genetic ancestry, right? So I think over the whole world, the amount of individuals that have European genetic ancestry is around 15%. So there's this huge dissonance, this disconnect between the fraction of individuals who participate in the genetic research, which again serves as the foundation of genomic medicine, and the worldwide population. So what this means in practice is that the findings related to genomic medicine that we make using research cohorts with European ancestry may or may not transfer well across ancestries or across population groups. So remember that before I talked about health disparities that disproportionately impact minority, racial, and ethnic groups. So the very groups that already bear a disproportionate burden of health in the US are the ones who stand to benefit the least, unfortunately, from genomic medicine at this time. That's the essence of the genomics research gap. So what we're trying to do with our collaborators from NIMHD, you also mentioned our work in Latin America, and our capacity building work is related to that, is really three things. So in my estimation, there are three things that need to be done to close the genomics research gap so that all people everywhere can enjoy the full benefits of genetic and genomic medicine, okay? So the first one is more data from diverse global populations. So we need to do a better job of recruiting research participants from what NIH calls UBRs, underrepresented biomedical research groups. And that includes here in the US and around the world. The second one is better methods to take advantage of this wealth of data that we already have to help ensure that the findings that have been derived from studies on European research cohorts can be transferred as best as they can to diverse ancestries. And the third one, and this is directly related to our work in Latin America and also in Africa, is building local capacity, right? So part of the challenge of the genomics research gap is the fact that there's a dearth of local capacity to do the kind of research in the communities that, again, stand to benefit the most from genomic medicine if we expand the reach of genetic research out to diverse populations. So we're working to build capacity in bioinformatics and genomics in Latin America through the Pan American Bioinformatics Network, and in Africa through an initiative called H3 Africa, Human Health and Heredity in Africa, which is funded by NIH and the Wellcome Trust.
Thank you. I have a few more questions, but I'll just ask the audience if you have any questions for the end. Please pop them into the chat, and we can get to those. So please feel free to do that. So my next question kind of pivots a little bit to the genomic medicine part of your research, which you've mentioned. You defined it, and I think you gave us a flavor of how it is currently changing medical practice with the rise of personalized medicine based on genetic analysis. What, if any, what's your favorite example of that? Is my first question, and my second is, what ethical concerns bubble up for you or this whole field that you see, that we need to pay attention to? Those are, I know, open-ended questions, but first, you have a favorite example, and then the ethical concerns.
Yeah, sure. So one of the examples that we are really interested in right now is the concept of pharmacogenetics. So historically, when you go to the doctor, you go to the physician, you present with some symptoms, they do their diagnosis, and based on their diagnosis, they say you have disease X, and we're going to treat you with intervention Y. And that intervention Y, for better or worse, nowadays is almost always a medication or a drug. Okay. Now, that traditional way of doing things does not take into consideration the genetics of individuals. And we know that based on your DNA profile, my DNA profile, the way that in which we respond to different medications can differ, right? Irrespective of our age, our weight, our sex. These are the things that we typically take into consideration when making drug treatment decisions. Irrespective of all those factors, our DNA sequence alone can really influence the way we respond to medications with respect to efficacy, how well the medication will work, with respect to dosage, how much we should take of any given medication. And what really, really motivates us is a big problem, which is adverse drug reactions. So adverse drug reactions are a huge challenge to the health system. And there are lots of people that have genetic variants that predispose them to have adverse reactions to different medications. So pharmacogenetics is the use of individuals' genetic information to tailor therapeutic decisions. In other words, to say, you should take this drug and not that drug, or if you take this drug, you should take this much of a dose. Now, that's at the individual level. We're interested in health disparities, and we know that although the groups that we're interested in, again, the racial and ethnic groups that bear a disproportionate burden of health and disease in the US are socially defined groups, there are genetic differences between those groups that are correlated with differences in genetic ancestry. And it turns out, for some reason, and we're still not entirely sure why, pharmacogenetic variants, those genetic variants that are associated with how individuals respond to medications, are variants that really differ a lot between population groups, including socially defined racial and ethnic groups. That means that medications that work well for one group may not work as well, again, on average, so I want to be careful here, for another group, and that information can be an important tool for the healthcare system, okay? And so let me just put a point on that. One of the things that we have shown is that we can predict for certain drugs, if you take a population of, say, 1,000 individuals, if you, this ties into the genomics research gap. So let me just take one step back and tie it to that. Genomics research gap, the vast majority of research is done on European ancestry cohorts. So now let's think about pharmacogenetics. We have a population pharmacogenetic profile based on European ancestry cohorts that says, well, the probability of an adverse drug reaction for drug Y is really, really low based on the European pharmacogenetic allele frequencies. But if you look at African Americans, for example, they may have a variant at much higher frequency than individuals of European ancestry that predisposes many more people of African ancestry to have an adverse reaction to that drug. And we've shown that you can have on the hundreds of predicted excess adverse drug reactions per 1,000 patients in African Americans compared to white patients, just based on the differences in pharmacogenetic frequencies. So that is an example of how we're using genetic information to kind of counter that genomics research gap and make better decisions that, again, hopefully will not exacerbate these health disparities that already exist. So you asked about ethics. Sorry, I know it was a two-part question, and it's a big two-part question, and I'm kind of glad that we're only left with 2 minutes to talk about ethics, right? Because we could easily spend half an hour talking about ethics. And there's so many different things to cover there. And one of the things I should say is that, I'm a geneticist by training. So I'm fascinated by genetics and I'm fascinated by DNA, but I'll just issue kind of on the ethics side of things, a caveat or a warning that genetics is not destiny. DNA is not destiny. Everything that I'm talking about here is probabilistic, right? So I started talking about, we are interested in understanding risk factors that elevate individuals' risk of disease and that may distinguish or may explain to some extent why different population groups have different prevalence of disease. All of these risk factors, including DNA, but also environmental risk factors, they're all probabilistic. In other words, they increase your probability of getting disease, but they are not deterministic. And I think that there's this erroneous notion that DNA is destiny, and that if you have this variant or this collection of variants, you are destined to get this disease. And that's definitely not the case. So I think on the ethical side of things, I'll just close out by saying DNA is not destiny. This is all probabilistic. We need to remember that when we're talking about these differences between groups in particular and what the implications of those differences are.
Thank you. We're going to hold you for a few more minutes because we've got a couple of great questions in the chat. So the first one says, are there specific genetic sequences that you have found that environmental factors would cause an epigenetic expression of type 2 diabetes? So are there specific genetic sequences that kind of you see as risk factors for type 2 that play with environmental factors?
Yeah, that is a great question. The short answer is no, although we would love to get there. So you're actually out ahead of where we are now. I will say that we have found what we call gene by environment interactions, but not with respect to specific sequences, but rather with respect to the genetic ancestry or the genetic background. So what we have found for type 2 diabetes in the United Kingdom is that for certain groups that have a higher level of type 2 diabetes in the United Kingdom, socioeconomic deprivation interacts with the genetic background such that socioeconomic deprivation is even more of a risk factor in that population. So it's actually has the effect of socioeconomic deprivation is actually exacerbated. But that's over the whole genetic background. I mean, you're a great question. Whoever asked that question would be really kind of the gold standard to hone in on a specific sequence or a specific variant, especially a site, let's say, that's DNA methylated or something like that. But we haven't got there. And to be honest, we're not even close. Hopefully one day we'll get there.
Thank you. The second question in the chat concerns risk factors that correlate with adverse ZNA situations. So the questioner asks, so you find a correlation between limb abnormalities, skeletal dysplasia, or childhood cancers based on zDNA? Or is it not zDNA, ZNA?
I don't know the answer to that, but I do know that when it comes to cancer, for example, I mean, I suspect that the answer is yes, right? It seems like an obvious yes, but I want to be careful because we have not found that in our own research. What I do know from the little bit that I know about cancer in that area is that cancer tends to come in clusters. Right now, the cancer clusters that we've worked on in our own lab are clusters that, are defined by ancestry. But there are certainly also environmental clusters of cancer that are related to the ZNA, right? The local environment, and especially exposures to toxins in the environment. So the answer to your question is certainly yes, although that's not something that we are actively investigating. Certainly yes when it comes to cancer. With the other limb issues, I just don't know the answer to that.
Thank you. And I'll close with one final question from me about pediatrics. So genomic medicine, health disparities, they are of obvious and particular relevance to the childhood population. Do you study pediatric diseases separate from everything else? And if you don't, is this a field that is not being as well explored as you think it should?
We do not study pediatric diseases separately per se, but I will say that one of the places where genomic medicine can really have a big impact in pediatrics is in terms of genetic risk. So earlier in the talk, I talked about the fact that we all have the genetic variants that we inherit from our parents. So while I'm not a genetic determinist, I do dream of DNA, right? And I dream of the day where every infant will have their genome sequenced, shortly after birth, and that genetic information will become part of their routinely used electronic health record. Now, how could that really pay dividends for pediatric medicine? Because you could define individuals' genetic risk of disease years ahead, especially for common disease that have big environmental inputs, years ahead of the environmental risk factors that individual is faced with. So you could really presumably intervene early and often for individuals who are identified as pediatric patients with high risk of disease to help, again, hone in on those modifiable environmental risk factors that those individuals should avoid. So I certainly think there's really great opportunity for genomic medicine, at the pediatric level with respect to identifying individuals' genetic risks early in their life and using that as a preventative tool.
Yeah, it seems to me to be especially applicable to the pharmacology that you mentioned, right? Drug reactions or adverse reactions to drugs. Boy, that's something I would want to know about my kids, right? As they progress through their childhood and their life, I would want to know, are they more likely to have an adverse reaction to a particular therapy for a disease? That would be, I think, pretty useful and not so controversial information to implement.
I totally agree. And you should know that there's no technological barrier whatsoever to having a button that your physician pushes for your child and says, I want to give this child this medication. Boom. Is it counterindicated? Yes or no? And in fact, in a class I ran a couple semesters ago, we had class projects, and one of those was a group that worked on pharmacogenetics. And what they did was they found, based on the DNA of individuals that they just got offline, what drugs were counterindicated for those individuals. So that technology exists, but it has just not been put into place in the clinic yet.
Wow. All right. On that very interesting note, we'll wrap it up. King, Jordan, thank you so much for your time, expertise. Really appreciate it. Thanks, everybody, for signing on, for those great questions. This recording will be available on the PTC website, I believe, and certainly we can spread the word because I'm sure lots of other people would enjoy it as well. So, King, thanks again, and we appreciate it.
Thank you, MG, and thanks to everyone who joined. The pleasure was all mine.
Take care.
Goodbye.